So we are delighted to have today in our Bar-Ilan University Vision Science Seminar, Professor Yoram Bonneh from our School of Optometry and Vision Science. Professor Bonneh did a BSc in Computer Science at Tel Aviv University and then an MSc in Computer Vision there. He then continued to a PhD in neurobiology at Weizmann under the supervision of Professor Dov Sagi, working on early visual processing. During his PhD period, he discovered the exciting phenomenon of motion-induced blindness, which was later published in Nature. After his PhD, he continued to do a postdoc at UCSF and Scientific Learning Corporation in Berkeley, where he worked on visual and auditory training and autism. He then did a postdoc at Smith Kettlewell Eye Research Institute in San Francisco, working with Professor Anthony Norcia on visual evoked potentials. Afterwards, he returned to Israel and did another postdoc at Weizmann Institute at the Department of Neurobiology, working on amblyopia and autism. He joined the Department of Human Biology at the University of Haifa as a senior lecturer, and in the last decade, he is at Bar-Ilan University School of Optometry and Vision Science, where he heads and leads the Yoram Bonneh lab that investigates vision and brain disorders. Much of his earlier research has been dedicated to lower-level visual processes related to spatial vision as contrast and contour detection, and these have been investigated in different atypical populations, including in non-communicating people. In recent years, part of his research is dedicated to studying non-communicating people by using eye movement patterns and characteristics, and especially saccades and microsaccades, including microsaccade inhibition, to reveal their underlying perceptual and cognitive conditions. He has many collaborations with researchers worldwide. He published dozens of peer-reviewed papers, has an H index of 32 according to Google Scholar with thousands of citations, and he also has 2 US patents in his name. So without further ado, we are delighted to have you today with us, Yoram, in our seminar, and we're looking forward to hearing about your exciting research, and thanks for joining us.
Hey, thank you, Sharon. I kind of feel at home because we are a few meters. Our rooms are next door, so finally I'm giving this talk, and the title of the talk is The Unconscious Eye, what involuntary eye movements or what involuntary aspects of eye movements revealed by brain processing, which is the work I've been doing in the last 10 or more years. It's not the whole work we do in the lab, but that's the main thing. And I'm going to concentrate on this, the majority of or all of the talk, depending how it goes. So I'm going to start with to tell you how I got to be interested in eye movement, because originally this was a very peripheral thing for me as a vision scientist studying vision. Was not very interesting. But then, I discovered or rediscovered this motion-induced blindness effect. Can you see it? Is it good enough?
Yeah,
So
definitely.
if you fixate in the center, you should be seeing the sum of the items disappearing, as long as you don't move your eyes too much. Okay, that's the some people claim that maybe what the movement is doing is kind of suppressing your eye movement, and then you have retinal fading. Now, we didn't, at that time, we did a study, actually, Tobias Donner and David Heger, together with Dov Sagi and myself, did an fMRI study where we found antagonistic responses when things disappear. So the ventral went down and the dorsal, the MT went up, and the signals were not the same as physical disappearance. So the impression was that it's clear that signal comes to the cortex, reaches the cortex even during disappearance, but still we wanted to test it, to rule this out, the story of microsaccades that would, or fixation eye movement that was suggested by Susana Martinez Conde, that microsaccades are actually countering fading. So perhaps what the motion is doing is reducing. So we did experiment something like this. Here the eye movements are superimposed of one observer, for example. And if you fixate, you will probably see the disappearance of the patch right here. The conclusion of that study was that we call it motion-induced blindness and microsaccades. This is together with Moshe Fried, Alex Cooperman, and Amos Arieli, and the other guys from the NYU. Cause and effect. Is it a cause or effect? And it was not. The conclusion is that microsaccades were not necessary nor sufficient to explain this phenomena. There was a modulation of microsaccade, as you can see. If you move your eyes, there's no disappearance, but that's not the explanation. There was another interesting thing there, that when there was a disappearance and when there was a reappearance, there was a modulation of microsaccade that was kind of similar to a modulation to a perceptual event. I'll get to it maybe later, but that got me interested in these tiny movements. So this is a recording of someone fixating, and you can see here the blue is the drift. I hope you can see it in nice refresh rate. The red is the microsaccades. The size of this effect is quite large. I mean, if you were sitting in a room and you use the rule of thumb, right, that's about 1 degree. So what you see here is 1 degree, right? This is 1 degree. So usually when you're sitting with people in a room, it covers their whole head. It's pretty large. 1 degree can be very informative. A lot of movement, but this looks like random. It is a kind of a random walk, the drift, but it's not exactly like that. And it's actually, when you dig into it, it's quite interesting. Just a remark about involuntary eye movements. I mean, we can blink voluntarily. We can even make tiny saccades, microsaccade, very small microsaccade voluntarily, but most of the time we are unaware of these movements and unaware of our blinking. And we are unaware of the freeze, the inhibition. So I'll show that involuntary aspects of eye movements are a rich source of information about brain processing in the normal and abnormal brain. I'll start with the basic, the inhibition. On the left, you see a stimulus flashing, and the observer is sitting in front of the screen doing nothing, actually. We tell them to count the number of patches, but they don't need to do any discrimination, anything, just count, just not to be, to fall asleep. What we see here is a raster plot. On the X, we have time. Each dot is a microsaccade, and each line is an epoch. Epoch that 0 is the time where the flash on the left appears. And you can easily see that there are microsaccades before, and then when after the stimulus appears, there is a complete shutdown. Here there is complete inhibition. And then there is a release of inhibition, very kind of coordinated, precise, across strides. And then going to some baseline. And you can quantify the raster plot by a sliding window. It can be a Gaussian or some more sophisticated kernel, but that's basic. Now, the question is, how does this depend on the stimulus. Just want to say that here that this is a very general phenomena of inhibition. Actually, we see it now even in the body movements, body inhibition or head inhibition, but you can see it in microsaccade, in the drift. I'll show it in the drift, blinks, microsaccade during pursuit, the catch-up saccade, and the pursuit itself, and microsaccade in free viewing can be generalized to that.
Yoram, you allow questions or to wait?
Okay, quick question.
Quick.
Yeah.
In many cases, it starts before the onset, before zero time.
Right. That's because it's regular and the subject can anticipate. So then the question is what determines the properties, onset, release, time course, and so on. So sensory saliency is a major thing. I'll show it in a minute because we studied it parametrically. Attention, there is a lot of work on attention. Initially, the thinking was that microsaccade are strongly related to attention. Well, there is evidence. Deviance, oddball, I'll talk about oddball. Valsecchi et al. Showed it in 2007. Anticipation in time. Task difficulty, we also have evidence on that regarding working memory. I'll show it later. Auditory cognition, we have also evidence on that. And language processing, we did some studies. And the general hypothesis is that the inhibition length is determined by the process time. So you stop the eye movement, inhibit them until you terminate some cognitive step or process. Now I want you to look at the figure on the left. I deliberately hide the legend here. Which of them, tell yourself, which of them is the high contrast and which is the low contrast, blue or red? Blue, you see a very strong, quick response and quick release of inhibition. And the red is slow inhibition and then some late and not very precise on time release. So if you thought that the blue is the strong contrast, you are correct. The blue is 25% and the red is very low. The way to think about it is obviously to go down to zero contrast, and then you would say that there will be no effect at all. So that makes sense that the red is a small effect. Now we wanted to quantify it, and the way we did it is, and this is not something that other people did. They usually analyze the rate modulation function, which is itself some kind of a computation on the raster plot. But what we did is to take the events themselves, the microsaccades, and average them. We call it microsaccade RT. For that, you need to pick a window. So we pick a first window is between 0 and around 200 milliseconds. That's to capture the onset of inhibition. Then a second window around 250 and 1 second to capture the release of inhibition. So if there is a microsaccade in the window, it gives you information. If it's no microsaccade, there is zero information. It doesn't have any effect. So you miss all the information of the absence of microsaccade, but that's not important here. We are looking for the evidence for time. So we do that and average the saccade, and we get a nice function, which is not exactly, but it's kind of a linear on a log scale of contrast. This is true for the inhibition onset and the inhibition release in a certain range. We do that's published in Jove 2015. We do it for spatial frequency, and we get something that when we're talking about the MSRT, the microsaccade RT, the release of inhibition. we get something which is very similar to the contrast sensitivity function. And in fact, if we measure threshold, this is shown here, and show it in z values, they really overlap almost perfectly, not completely, but almost perfectly. So this is the latency of the microsecond, the release of inhibition, which seems to reflect how much time you need to complete the process of the initial perception of the stimulus is related to contrast sensitivity. Remember that contrast sensitivity is measured as the contrast detection threshold. Here we measure the response of the system for high contrast stimuli, not very high, but high contrast stimuli. Okay. Now, what about eye blink? You would think that they are nothing, they are not very interesting, they are there to moisten the eye and so on, but it turns out, first, that they are entrained. If you plot it as percentage of change, then they are completely entrained to the stimulus. Now, that might not be too surprising because we ask the observer to count the stimuli, so they might not, they might take the idea that not to blink when the stimulus comes. However, when you compute the exact time, the blink RT, what you get is something that is almost identical to the microsecond RT. So here we show the blink RT on the X, the microsecond RT on the Y. These are group averages, the function for different contrast levels, and it's pretty much on a line even in terms of the millisecond. And the whole range is typically of like 50 to 100 milliseconds, spanning the whole contrast and spatial frequency range. Now, blinks are not very frequent, maybe one in few trials, but and microseconds are a few in a second, but you don't need a very sophisticated eye tracker to measure blinks. Now, if this is so general that the blinking is the same, so maybe it generalized to other things, like microsecond inhibition during pursuit, inhibition of pursuit itself, drift inhibition, and microsecond inhibition in free viewing. That's a generalization that I'm going to explain later. This is a paper published by Balziv and myself in June 2021. The catch-up saccades are inhibited. The pursuit itself is inhibited. Is a little bit noisier than the microsecond in fixation, but it works, and we can compute the dependence on contrast, and the spatial frequency is very similar. The ocular drift is also inhibited, but measuring drift requires high precision tracking. And Ehud Achisar and Liron Gruber did a paper 2020 with, they managed with the iLink, though there's a lot of criticism that iLink might not be accurate. Managed to show that there is stimulus dependency and functional significance of the drift. It's not just random. Rucci and colleagues did also show very nice demonstration. They have a very precise eye tracker. So what we did is to team with a group in Poland, in Nicolaus Copernicus University in Torun, Poland. And they do a retinal tracker, freeze eye tracker, we call it. So retinal tracker, it's a kind of a scan laser ophthalmoscope, imaging the retina, the blood vessels in the retina, and so on. And they could get a very, very nice precision. And the sample-to-sample RMS is very low. So they recorded for us, and we did analysis. And what we see here is an example how the drift is slowing down, and in fact, the eye became more concentrated in one place, which you can see in this video. And Here you see the comparison of drift inhibition to the microsaccade inhibition. The drift inhibition was assessed in BAL, did it in 3-4 different ways in diffusion and different methods, and velocity, area of the drift, and so on. And we could get some result that is related to the contrast and special frequency. This is how we had difficulty to publish this. This was a couple of years ago because the device was not proven yet, but I think that now they published a paper on the device and we'll get a green light to go with this paper. Okay. Now, I want to suggest, this is a work of Oren Kadosh. We together looked at the idea that previewing is in a way similar to flash stimuli. So if you are doing a saccade, a big saccade, greater than 2 degrees, for example, you land in a certain place and you get a flash to the visual cortex. Hamutal tested this with monkeys, even with microsaccade, there is a flash of a big flash on the visual cortex. I'll show it. And does it behave the same way? I mean, you move your eyes, you make a saccade, and you land on a new place, you get a flash to this visual system. Now you should inhibit your blinks, your microsaccade, your drift, depending on the stimulus. Okay, so we had observed just free view things like that, high frequency, low frequency, and we looked at the microsaccades after the inhibition. So this is what happens with flash stimuli, and this is what we got with a similar analysis triggered by the saccade. We did another study with fixation-related potentials, but that's not in the scope. I don't have time to talk about things that are not eye movements in this talk. We could get the contrast special frequency response function from free viewing when the same analysis, microsaccade inhibition following the big saccade. Okay. So this is a good time to stop and ask what is the function of the inhibition.
May I ask a question about that previous one?
Okay, good. Okay.
Because did you see any difference depending on where the saccade landed on those grids? So whether it landed on one of those gabors or between gabors or at the edges of gabors?
We didn't analyze that. It's pretty dense, so statistically you are most of the time on a gabor. But it's a good question. We could analyze, but it's a little bit of work. But it's a good.
Okay.
So what is the function of the oculomotor inhibition? So it makes you are processing previous stimuli. That's one idea. You slow down the drift. Maybe it's an issue. First, one question you can ask: is the microsaccade a really big thing for it's a small movement. Is it a big thing for the cortex? So Hamutal, Slovin, and her colleagues just published a paper looking at the effect of microsaccade in the primary visual cortex, even when there is nothing on the screen, blank screen. So, and Hamutal can correct me if I make mistakes here, but even if there's nothing on the screen, so you move your eyes to make a 1 degree microsaccade, and there is extraretinal input, must be extraretinal input. They looked at the voltage-sensitive dyes imaging, high speed in the V1. So interestingly, the whole fovea area got 100 milliseconds after microsaccades, it got synchronized. Before that, you have, if you look at the response maps, you get a suppression and then increase of response. Both are interesting. I mean, this is just a microsaccade or on a blank screen. There's nothing. So if this is correct, then the microsaccade might be a global signal for the visual cortex and the brain in general. It's a signal event, and you could think about it as, and that's the hypothesis I want to suggest is that the oculomotor inhibition synchronize eye movement with perceptual and cognitive events to enhance them via sensory action integration, like dancing in sync with rhythmic music that enhances the rhythm. You create actions that are in sync with the events, and this enhances the events. The oculomotor inhibition, the OMI, is typically terminated at the end of a processing stage, thus reflecting processing time. So if we measure this time, we get interesting measures like those I showed you, for example, the connection to contrast sensitivity, to spatial frequency, and so on. Okay, so now I want to ask the following question. A surprise in time, I mean, there are a sequence of events and something surprises you, is a salient event. Would it result in a short OMI like high contrast, a short inhibition like high contrast? I mean, this is a salient event like high contrast, you have very quick inhibition, you press the brakes, and then you have a strong response maybe to light your V1 with a big quick microscope. So we ask this question in the following way. So this is the sorry. Okay, I'll skip that. You can imagine what I wanted to show you is this. Maybe I have it after that. Just a second. Yes. So the approach was to have observer just look at a sequence of events of two kinds. For some reason, it's all green, blue one now, but now you have a red, and it's random order. So you might have a few reds, blue, and so on. When there is a sequence of red, the blue is a surprise. So here's one way to look at it. Imagine that this bar is time. You move from left to right with yellow, and suddenly it changes to red. And if you assume continuity, which is something that the brain does, this is a surprise. In space, this is an edge, a change. It can be done statistically. You have some reds here, but basically this is mostly yellow, and you get red here, and there are some yellows here, but it's basically a surprise here when the red comes. Maybe it's not as big a surprise as here, but it's a surprise. You can do it with discrete events, same idea, and you can do it with complex patterns. And the simplest sensory prediction is continuity. And the question is, so now we use this as an example of surprise and ask whether the inhibition, the OMI, will be longer or shorter. Okay, and that's the experiment we were doing, and we tested different variations of stimulus. This one is a contrast. We have high and low contrast, mixed in random order, 50-50%. And I'm going to show you data from one observer first. Here we compare the rate modulation function of microsaccades between of a high contrast patch that was preceded by a sequence of low contrast patch in red. And that's a change, a surprise. And high contrast patch preceded by a sequence of high contrast patches in blue. And as you can see, there is a shift. Actually, there is a delay. The answer is, surprise, increases your inhibition. It's longer, prolongs your inhibition. Here's the Russell plot. You can actually see the Russell. This is a single observer. You can actually see the shift in the Russell plot. We can use this microsaccade RT calculation. There is some outlier here for some reason, I don't know, but we plot it, and this is a new trick. We plot it as a function of the sequence of preceding events. We denote the sequence below here. The last one is always 1, meaning that the current, and it's going from bottom to up. So 1, 1, 1, 1 means that the current was preceded by 3 similar, the same contrast events, and 0, 0, 0, 1 means a change. The high contrast was preceded by 3 low contrast patches. This could be described as a serial dependency, and it, without this outlier, it really goes on a line, and I'll show you a lot of lines like that next, but this is a single observer. Another way to show it, a different way, but sometimes it's clear, count the prior event separately, either different here or same. So this is 1 or more, 2 or more, 3 or more identical events. There is a speed up. 1 or more, 2 or more, 3 or more from the other kind of event is slow down. Surprise is a slowdown. This is the work of Yael Schwartz and myself, and it actually started long ago, but this is a new version of this work. So this is a kind of serial dependence, and well, I can tell you that the freezing is proportional to prediction error. I'll talk about it in a minute. Here's a bigger picture. We do it for contrast. We do it for house faces, houses, color, and position. Position, there is a little dot here. This is actually the TOVA, the TOVA test for attention. The dot can be high or low, and what we get is this serial dependency line, as I've shown you before. It's noisy, that's true, and the effects are not huge, but they're very significant, especially here because we have 34 observers, so we have a very nice effects. And in both ways of plotting it, the pattern is always very similar. A slowdown for a change, a speed up for similar thing. So repetition speeds up, as a change slows down.
Repeat the binary code down there, the 0011, what do you mean?
The coding, the binary code here?
Yeah.
Okay, so it's arranged like a binary code. 0001 means different, and the last one is this one, the current. Current preceded by 3 different. On the left, current preceded by 3 same. In between, this one, for example, is current preceded by, if I go further away, same, and the last one is different. Just before one is different. So what this implies, if this really goes on a line, it implies that, like a binary code, that it's like exponential decay. I'll show it very briefly in the next few slides. This is diving in for the TOVA data. It's kind of nice data. So this is the history, all the combinations. Pretty much goes along a line, not perfectly, unfortunately. If we go just 2 back, it really goes on a nice line. What goes on a nice line? On the MSRT, the microsaccade RT. Minus 20 millisecond plus 20 millisecond. Okay, it's just a bias that's sorted when you sort the data. You get this is slower, the other is faster. Okay. And if this is really going on a line, the implication is that it's 1/2 to the power of n weighting. I have one slide before just talking very briefly about a very basic, simple model, but just want to show you that it's actually working for the blinks, which is very surprising. Well, it tells you why the term "she didn't even blink" means that she pretended not to be surprised. Okay. So here we show the same thing, the serial dependence as a function of this kind of binary code, and the other type of stimuli. Here we had the target and destructor. In this case, it's a special case, but I don't want to spend too much time on this unless you ask me, or ask at the end. I can go more. And a simple model means that the freezing is proportional to prediction error. And how can you predict prediction? You actually predict the probability of the next one to be red, for example, or to be high contrast. How do you predict that? You assume that the probability should be according to the recent past. But the recent past, you take it with exponential decay. So you do this and you can get very similar results as the model. It's very, very simple model. And actually, this kind of behavior we find also in psychophysical data, and we do a big thing out with it with Dov Sagir and Noga Pinchuk. We analyze a lot of psychophysical data from the past that look like that, not the microsecond RT, but the percentage of correct responses. One last comment about this is that it is about predictability, because when you do something predictable, it's not just changes. It's not just the physical changes from a house to a face. 2 faces, 2 houses, 3 faces, 3 houses, whatever. But in this case, this is totally predictable. And if you do this experiment, you don't get the effect. If you do the unpredictable, you do get effect. But yeah, okay, that's one thing to say. If you have a quick question on that, that will not pass my time, because my time is limited. So maybe we'll better leave to the end. I'll do that briefly. Familiarity. So I don't know if you are able to see this in proper timing, but if you do, you might see a universally familiar face hiding there. This is a backward masking. Actually, this is 2 masks that follow the universally familiar face. We do this experiment, and here are actually it's like a lineup. There are 8 faces. 1 is universally familiar, 7 not, and we get both for the blinks and both for the microsecond. We get, this is the work with Gal Rosenzweig, published in Scientific Report 2019. So we get the prolonged inhibition for the familiar face. We can quantify it, and highly significant. The MSRT, the microsecond RT, is longer for the familiar and beats all the other saccades are better than blinks, but this works too. So we took it to something practical. Can we do a lineup with like people do in the police to detect concealed information? It's called concealed information test. We did a phase. We did some reference. We tested the observer. Okay, we let participants. We told them that they are doing a mock terror experiment. They have to choose a number, and this guy. They see some videos on this guy, learn the name and the city. They're going to kidnap this guy. Okay. After they learn about this guy, they are tested passively viewing for 20 minutes. And we need, so we have the name, and we have 8 names, 8 city names, eight faces. We need to find which is the one that they learned, that they have chosen. And we did this with the microsaccade inhibition. Actually, we took several measures, not only the microsaccade inhibition, and we computed not just the MSRT, but more deviance, and we were able to develop the CIT, a continuous information test on individual oculomotor deviance, and we had 100% accuracy in identifying which one is the one they chose, and 95% to detect which one were innocent. There was a group of innocent that didn't participate, and we had to detect whether they are really innocent. Okay. We did another study in free viewing. So we were really interested in how this applies to natural vision, not only flashes. So if you are, here's what we did with Oren Kadosh, this published in Scientific Report 2022. You take 4 seconds to inspect a face. Each saccade on affected face is pretty big. It covers a large part of the screen. If you do a saccade, you get a flash on your cortex. It's like a flash in a psychophysical experiment, and then you hold your saccades, and can we tell by this kind of inhibition whether the familiar or not familiar? And it's not as good as the MSRT, but of flash stimuli, but we could get the difference for the unfamiliar. Well, there's 3 outliers here. These are observers. We did the same with fixation-related potential, but that's a different story, and I'm not going to talk about it. Okay, now I'll tell you kind of briefly about some other oculomotor inhibition studies. Language is one. We did in 2017 with Michal Ben Shachar and Maya Jablonski, a study where we showed text words, and some words are real words, and that's in Hebrew. Some words are real words, and some words are non-words that are similar to real words, but they are not real words. And some words are non-words that are not similar to real words. And we measure the microsaccade inhibition, the MSRT, and we get this effect. Real root, that's similar to words, is different from inverted root. Recently, we did the same with spoken words, hearing. Now, this is very tricky because and very difficult to do because what is the real onset of the word? But it turned out that with proper, that's Oren's work, with proper analysis, you can get it even with the blinks and the pupil RT and the microsaccade RT. I think I showed it here. So we can pick up the processing steps of this language decoding, where it takes you longer when a non-word is kind of similar to a word. So it takes you longer to reach the decision that this is a word. Another example that's Vincenzo Rizzuto, an Italian MD who was here for some time. We inspected music. I want to show you. Can you hear that? This is a Bregman classical grouping experiment. Can you hear that? Yes. Okay. Okay. So okay, go. So this is what you see: low, high, low, and when it's too fast, they merge together into one. You either hear the low or the high, and in between, you can actually select what you hear. Would the eyes sync with what you select? And the answer is yes. It's not a huge effect. But we are doing spectral power in order to figure out, but actually the bottom line is that the eyes are synchronizing with the perceived. So actually your choice what you want to attend, and if you perceive the high tones separately and you are attending it, you'll get a different frequency from the low. Okay. Okay, and finally, it's working memory. It's not really, we hope that it will be in the microsecond inhibition, but it's more microsecond inhibition latency, but it's more actually, if you need to remember 3 items, you have more inhibition than if you need to remember 2 items or 1 items. And the items here are presented briefly. Always 3 items are presented, but you have a mark here to show which one you need to remember, and then you will get another one that you need to say if you have seen it before or not. Okay, so we will get this. So I have a little few minutes to talk about autism, what we do with autism. I'm interested in that for some time. I did a study with this guy, nonverbal autistic, in 2008. Actually, that was a few years before. These people cannot speak. So the question is, and they are 30% of the autistic spectrum. They are minimally verbal and typically fail the cognitive test and typically behave as if they don't really understand what you're talking, or they don't really understand the situation or the environment, whatever. But this is not the whole story with them. And the hypothesis is that severe autism is characterized, at least partly, and at least in some of them, as an output problem, a core action control deficit for voluntary behavior, which calls for the use in involuntary behavior. So what we did initially, that's a study of current alert, but I'll tell you, initially we tried the microsecond inhibition. We got results with one observer to kind of hidden knowledge. What is the question? What is the capital of France? Does he know it? And you get longer inhibition for that, or a familiar name and we got it. But that was very difficult and very noisy, and this didn't really work well. And you need a very precise eye tracker for that. So we ended up doing something with a simple Toby tracker with this paradigm. You need to you read a word and you have to go with your eye gaze to the correct picture. You can try it yourself. What you see here is a recorded eyes from one observer. It's pretty easy, actually. We call it cued looking. You get a cue and you get the idea, and it's almost automatic. It's not completely involuntary, but it's and this is non-stop. You don't wait. Just put the nonverbal autistic in front of it, and you want to see what it gets. And some did, actually, most of them did right. Here we plot the eye gaze position when the stimulus was on the right side. When the stimulus goes on left side, it should separate like this. And this guy on the right is not okay. And there were many of them that did okay. We tested 30. These are 20 of 30 that I show it. If you look at the correct trial, they are very similar to control. And some of them did 90% correct, but not all. Now, dramatically, when they did pointing on cards, they did pretty bad, 60% on average. So you have some people here, some people here who are 90% correct on the eyes, but poorly on pointing. That's a diagonal plot to show. Okay, now this is about knowledge, about knowing to read. But what about simple things like this audible paradigm? This is the work of Thomas Hass. We were looking. We initially asked nonverbal autistic children to point to these audibles. Pretty easy task. Here's an example of a child that his eyes go correct.
Amen.
But look what happens when he points. The eyes initially went correctly, but then the hand goes to the wrong place. And in this case, the eye gaze was by far better than pointing. And the group effect, I'll show you here. We found this effect, it's not as dramatic as in this guy, but other children show this as well, which is pointing at chance level and the eyes goes correctly. Another study, this is a totally different study, we looked at the randomness of eye movements in free viewing. That's the work of Inbar. We took data from almost 200 autistic and 50 controls, something like that. And we analyzed the gamma distribution of the intersaccade time interval. This is an idea suggested by Elizabeth Torres for randomness of movement, but we took it to the intersaccade time interval, and this separated this cohort very nicely. The children just watched a movie clip of 1.5 minute. Okay, we can extract better AUCs if we take different parameters. And it's also correlates with the severity of autism. The last thing I'm going to show you on this is the entrainment. So this is the work with Shlomit Becker. They did an experiment on 30 autistic children 6 to 9 years old and 20 controls. There is a stimulus, cue, that flashes 1, 2, 3, 4, and then there is a beep, and they need to press the button as fast as possible. Shlomit and Becker and Sophie Molhon and colleagues recorded EEG, and they found less intertrial phase coherence. But we took the eye movement, and the first thing to look is microsaccade, right? So we see this entrainment. There is a flash, there is inhibition, release, and so on. And then there is the beep, and you need to press. But what happens in this entrainment? Is it the same for the autistic and controls? Well, not exactly. You don't see it with the rate modulation, but when you do the MSRT, you see that for the control, the MSRT is becoming longer. Each event is getting closer to the target, then they need to take it more seriously. And the variability, which is a marker of entrainment, goes down. This is the standard deviation of the MSRT, the microsaccade RT, it goes down. But not so with the autistic. And same with the pupil, but that I don't want to dwell on the pupil. Okay. Nice. So I could actually show you more things, but I'll leave some time for questions. So we have new directions. It's more stimulus-driven, the biomarkers of brain disorders. So TBI, ataxia, CP in children, this is the work, and psychosis and aging, we can do that. So we do something like this, simple tracking. A lot of people have done this, but we do it with the occluder and with trials. So we don't have pursuit that is continuous, but it resets and starts every time from the beginning to out, from center out. So we can get a lot of parameters with the traumatic brain injury. For example, the occluder-induced deviation, the pupil dilation when you start the trial and do the catch-up, and how smooth the target is. We have a lot of data on all of these disorders that I mentioned, but I'm not going to show this here. Just to show that this is a work of Shimrei Shani. There is a nice correlation with the clinical, this is functional independence measure. So different measure, including verges, for example, are highly correlated with the condition of the subject. And I think that's it. Okay, thank you.
Lovely.
I have another one. Wait, no, okay, I don't have it here. Okay, I wanted to show you the eye movements of all the subjects, but I missed the slide. Thank you.
Thank you very much, Yoram. And I want to invite everybody to unmute yourself and let's give Yoram a big applaud for a lovely and interesting talk. And I want to open the floor for questions if anybody has any. Ehud.
So what do you think is the function of microsaccades in vision based on these results?
So, okay, so in a cognitive process and perceptual process, you have events. The microsaccades enhance the events because they generate a global signal to the visual cortex. So that's it. It's like the dancing analogy, the metaphor. So if you move, if you listen to music, you're not allowed to move. If you're listening to music and you're not allowed to move, the impact of your music will be smaller. You want to emphasize, it emphasizes the events. Inhibition, you don't need to blink or to move when something is on the screen. You need to not to miss it. But
you show very clearly, very systematically across many cases that you
sorry, I missed.
During processing.
Oh, maybe my or yours.
Inhibit the microsaccade, right?
Sorry, there was a disconnection here, or I didn't hear. Can you repeat this?
I'm saying what I get from your systematic description. You say you read many tasks. In all of them, and by your claim, is when the processing takes longer, the inhibition of microsaccades takes longer. So during the period that is important for the brain to perceive, it inhibits the microsaccade.
Right. That's
true. It simply says microsaccades disturb.
Right. This is one part of the story. Second part of the story, when the time ends, you want to do a movement. There is a very precise timing of the movement. When the time,
of course,
the inhibition
ends. The inhibition ends, and then you have some rebound.
Yes, and this could be functional.
Yes, so this is a great marker. I agree with you. It's a great marker for processing time, but it's important to perceive microsaccades are inhibited.
Exactly. Yeah. This is the one part that the other is sure.
They're not part
of the story. Yes, I agree.
Michael, do you want to
Yeah. I don't actually have a question. I just want to say thank you for a very interesting talk. Lots of new stuff for me. Thank you. That was it.
Thank you very much, Michael. Thank you very much.
I actually have a question related to maybe, I don't know if it's like a contradiction or things that are, if you take into account the contrast effect on microsaccade inhibition and the surprise and maybe going in different directions. So I want to ask if you think that it could be that, for example, when you have a surprise event, you are stopping because you have predictions that more surprises are going to be coming your way. I mean, you have to stop the senses and you have to let more information more information sink in, so you don't want any disturbances. Whereas with the contrast, it's already like happened. So I'm just thinking whether it has to do with past and potentially future aspects. I don't know. Maybe it's
The inhibition for the contrast is sensory. The contrast, high contrast, takes shorter time in the perhaps in the
yeah,
retina, in the visual cortex. Everything, the whole transduction is faster, and you get the event, and it's a
yeah.
strong event. That surprisingly, you need to update your records to not to make these mistakes again. I mean, you make predictions and you take the prediction error to that's automatic. You need to update all the time in order to reduce the prediction error to the minimum in the future, something like that.
But also, in everyday life, when you have surprises, say you have a weird voice coming, a weird noise or something, you anticipate that there's more to come. So you have to keep your senses. So there's
So
some
you
prediction,
immediately
not only of the
maybe,
prediction error,
yeah.
but you're predicting something that, in your like semi-random sequence, these were very localized events, and they were not, self survival.
Right.
But
So
say
then
you
what
have
happens
some,
if the tiger appears? So
yeah,
you
or if
freeze.
you think
First you
there's a
freeze.
tiger, you want to be, you want to have, I'm just wondering whether it also has to do with what you're foreseeing for the future, not only just a thought. It's not that, but I'm just thinking maybe that's why it's for longer, because you are anticipating that maybe something additional is going to be
I
coming your
see what you
way.
mean.
It's not that.
Yeah, so what we find that the duration of the inhibition is proportional to the how big the surprise was statistically. If you were certain it should be red because you had 100 reds and now come blue, it's a big surprise and you stop a lot of time. If not, these are tiny surprises. What happens with big surprises? I don't know. I mean, yeah, maybe you prolong the inhibition to
Okay, and I'm just a
ready for
thought.
the
It's
tiger.
just
Yes,
I wanted to
Hamutal,
ask,
I see.
yeah, I wanted to ask a question. I think I want also to relate to Ehud's question. I think there's an interesting relationship between microsaccades and the fact that you have later on an inhibition. As Ehud mentioned, when you start processing, it seems that there's inhibition. So in a sense, it contradicts the role of microsaccade in the sense that when you are processing, there are no microsaccade. So then the question is, in that sense, what is the role of the microsaccade? And so I have one question which I will ask in a second, and I wanted to refer to this and address Ehud's question. From what we see in our studies, microsaccades are actually recruiting certain brain mechanisms that later on enables better processing of the stimulus. So for example, there is an attention that is linked to microsaccade. And when a microsaccade is executed, one can actually follow the attentional mechanism that is linked with the microsaccade, which is then directed to the stimulus. And then there's time for the stimulus processing. A similar,
well,
yeah.
but just to make sure,
Yeah.
you're talking about microsaccades that are voluntary, perhaps, or
No,
they're
it
just
can be voluntary, but we
but
looked into
not
spontaneous one, yeah.
Okay, this is
Yeah.
spontaneous. Okay.
Yeah. And also the anti-fading thing. What we can see that when microsaccades are performed over or in relation to low contrast stimuli, then the stimuli are enhanced and then you have time to process them. So in my view, actually these two things are going together, right? What Ehud actually referred to and actually what you see, because the microsaccade recruit the brain mechanism the additional CPU, whether this is attention or whether this is anti-fading effect. So refreshing the neuronal activity of the stimulus, and then you have time to process it
But this is
without
after
any additional interferences. This is at least part of what I think is happening.
But this happens at the end of the processing, so to speak.
No.
So at the end,
No.
you have this burst of microsecond.
No.
At the
So, for
beginning,
example, yeah.
it's the beginning and the end.
Even in the beginning. At the beginning, you don't show. You show.
Oh, but Hamutal talking about the function of the microsaccades
Yeah.
to refresh.
So suppose that you have a stimulus which is, for example, low contrast, or which is presented somewhere there, and you show the stimulus.
But
The
it does
animal
not
can
improve
shift
microsaccade.
attention. Yeah.
But did anybody see that showing a stimulus increases microsaccade? I see only inhibition.
Yeah. Later on, yeah.
So maybe at the end, it marks the attentional flag that you mentioned. At the end,
Yes.
you say, okay, I'm done. Go on. Something
Not
like
exactly done, but additional processing. This is what I think that is happily acting. So it looks like the microsaccade might be recruiting additional brain mechanisms. So that's interesting. And what I wanted to ask Yoram in that sense, Yoram, how do you think that the oculomotor inhibition process that you refer to, what do you think, how do you think this actually occurs in the brain? Which nuclei are involved? I mean, how is it done?
I don't know, actually. What I can say, it's very general. I think we see it in even in the head inhibition. So maybe it's something completely, you inhibit your head. You're moving your head all the time, and when there is an event. So, but I think you are the better person to answer this.
this data of Hafed that shows that the supercolliculus is.
Oh, you mean the
It's
control of the
microsaccadic.
yes, the endpoint, but a lot of other regions are involved. It's not only, I don't know. You think it's only superior colliculus? It's a long. I mean, this is
No, not only, but this is kind of the hub. So the kind of
it's a hub. Yeah, that's right. That's true. Of course,
it links head motion and saccade, like all these surprise things, all these reactions. Supercolliculus should be the hub.
Yeah, that's true. But we're talking about much higher processing involved and sending some top-down signals for this inhibition. I mean, it depends on what can things that are more higher level surprise. Something is familiar to you. You see a face, and suddenly comes a familiar face. It takes more complex processing to detect this, and this goes down to the inhibition. If the face is familiar, for example, or if the face is something, that's the idea behind the concealed information test. Once you see the familiar face, that's a big event, and you freeze your eyes.
So it has to be a region that is highly connected to many high-level cognitive, memory, and motor. So severe colliculus certainly sounds like part of the network that may be related, but it's probably something also subcortical in addition, maybe not only.
I think the physiologists are the ones to answer this. I'm not an expert so much in this. Sorry.
Well, another point, you say freezing the eye, but it's not really freezing the eye. You inhibit microsaccades, but the drift is there.
Term the freezing is the term coined by, I think, Martin Rolfs gave this freezing idea. It's a relative. I don't know. Freezing is complete. It means complete.
It's freezing, but you have the
No, it's just a term to describe a reduction or inhibition to certain.
Misleading term, because freezing meaning no action. but actually you have the drift
Some
collecting
freezing,
data.
reducing of the action.
Reducing the disturbance. You're reducing the disturbing factor. It's not freezing. I wouldn't call it freezing.
No, but that's not, I didn't, but I think it's a reasonable term to talk about freezing. I don't know. But inhibition also is not total, it's statistical.
Yoram, thank you so much for a really interesting talk, and I'm sure everybody's going to have a lot of things to think about following this and be more aware of their freezing and inhibition aspects. So I want to thank you so much again for joining us and for sharing your research with us. And I am not yet sure about next week. But we will have a talk in 2 weeks for sure, and we will distribute the information hopefully soon. So thank you everybody for joining us, and thank you Yoram for the wonderful talk, and best wishes everybody.
Thank you.
Thank you.